Neuroscience at Hopkins School of Medicine and the Simval Krieger Mind and Brain Institute, of which she's also the scientific director. Her research focuses on the function and development of higher level visual cortex. Dr. Nielsen earned her PhD at the Max Planck Institute for Biological Cybernetics in Tübingen, Germany. There, she conducted research in the lab of Nikos Logothetis, investigating the encoding of objects in the inferotemporal cortex. And in 2006, she joined the labs of Ed Callaway and Rick Krauslis at the Salk Institute in La Jolla as a postdoc. And her postdoctoral work focused on two techniques that allow the study of the function of neural circuits in vivo with a high degree of specificity, which is the viral vector-based approaches, and two-photon microscopy. Dr. Nielsen joined Hopkins in 2012. Ongoing research in her lab focuses on the structure and function of circuits in higher visual cortex, as well as their development and plasticity. So, Kristina, it's such a pleasure to have you. Thank you for making time to be with us. And of course, after her presentation, we'll have a Q&A, and we will have also an intervention from Dr. Chris Porter and Giancarlo, our very own Giancarlo Molina, to talk about training programs at Hopkins. So, Kristina, thank you.
All right. So I will share my screen. I hope you're seeing that. Okay. Okay. So in general, I can only see, some of you will see how well this works out. But if you have questions, please just interrupt, raise your hand. Maybe somebody can tell me if there are questions. I'm more than happy to stop and address anything that's unclear at any moment in time, much rather than going on and talking about stuff that goes over everybody's head, okay? All right, so what my lab in general is interested in is vision. And so what I'm going to start with may make you laugh, but it's actually true. Namely that vision is your superpower. It's all of our superpower. It's one that we take for granted, but it's one that we still have. And you might wonder why I could be saying that. So let me give you an example. So look at the slide. One of our very talented graduate students, Alexandria, made the slide in a software called Blender. So there's no way that you have seen this particular scene ever before. But I assume it still basically instantaneously makes sense to you, right? You can look at this, and not only do you know that this is a scene that has different walls, there's all sorts of information that you get from it, right? So you know that it's full of different objects. You know what materials these objects are made out of, right? You know this thing is out of wood, that's glass. You know not just the material, but you know how it would feel like to the touch. So you know how the wood would feel like when you touch it. You know that this is probably soft. You know that the glass will be cold and smooth. You can make predictions about the future even, right? You know that if you toss this glass sculpture around, it would probably shatter. The same would happen for the vase. You know that if you cut the strings for the balloon, they would float away. You know that this little thing is about to fall over, but it probably would land softly. You know that if you kick this basin with the liquid in it, that it would slosh out. All of that by looking at a scene that you have never seen before. In addition to the things that I've talked about, what vision allows you to do is to assess things like value, to assess things like health, to assess things like emotions, how somebody is feeling, right? So all of that, I think, justifies the statement that vision is our superpower. Another way to show it is that basically, as you look around a scene, it's almost like this image that comes from the BBC TV series of Sherlock Holmes that I hope all of you have seen, in which Sherlock Holmes, whenever he looks around while he's reasoning through a particular case, all of these labels are almost like steaming off the visual world around him as he's thinking about how to solve a particular mystery. And the same more or less happens while we look around us, right? There's so much information that you can derive just from looking at something. The ironic thing is that we ignore vision because it comes so easily to us. And at the same time, it's one of those things that computationally is actually really hard. Computer vision by no means is as good as human vision. Artificial computer programs have made a lot of progress over recent years, but they still don't come close to what you take for granted every second of your life. Which is even more ironic because the things that we consider to be really hard, like making decisions or learning particular games, those are actually easy for computer games, for computer systems, right? There are now networks that can beat even the best players at Go. But again, something that's as simple to us like vision is really hard for computer programs. And another thing that, just as a food for thought, is similarly hard, that we similarly take for granted, is walking and somatosensation. So how you grasp something, all of that is also very simple to us and very hard for computers to achieve. So my lab generally is interested in this, in vision, but more specifically in understanding these complicated functions, like how we perceive objects. And we usually study that in adult animals. But we have, since I started my lab at Hopkins, also gotten interested in how all of these functions develop after an animal or a human is born. And it turns out, while we already know sort of a limited amount about how these higher level visual processes work in adult animals, we know even less about it, how they are set up during development. So generally, what we know is that, and this is mostly based on observing and how babies and like toddlers and then older kids look at scenes. So we know that our vision over time gets better. This is sort of a simulation that shows that basically newborns have very blurry vision and very poor color vision. They also have perception only at a very limited depth. And so then as you get older, you eventually end up with this really sort of clear and good vision that you are used to. The other thing that we know is that obviously the brain changes as we get older, and that, for example, the number of neurons changes over time. But nobody has, at least when it comes to these more complicated visual processes, nobody has tried to put the things together and to actually figure out what happens in the brain as your visual system is wired up and as all of these complicated things that I just talked about are set up. We just simply don't know how that comes about. And so we also don't understand how things could go wrong. What happens if somebody's blind from birth? What happens in developmental disorders in which vision seems to be impaired? We just simply don't know how that could come about. And so that is one of the big questions that my lab is interested in. One of the reasons that we know so little about how vision develops is a simple lack of animal models or general models. So in humans, it's in generally already limited the kinds of techniques that you can apply. And then so in general, you have techniques like EEG, you have behavior, or you have fMRI, which all allow you to measure brain function to some degree, usually at a very coarse level. But the problem with development is that the age groups that you're likely most interested in are simply not willing to cooperate with you. So try to convince a toddler to lie still in a magnet while you're scanning their brain. That's likely not going to happen. So there's a real limit in how much you can use humans to understand how higher level vision develops. Usually, monkeys are the standard animal model in which we study higher level vision, and they're enormously useful, and they are sometimes used for development. But they're simply really complicated, especially if you need larger cohorts of animals because you're interested in understanding, for example, how changes in development impact the visual system, or because you have to deal with natural variability in development. Usually, developmental studies in monkeys are limited to very, very few animals that you can sort of follow longitudinally. Mice are everybody's favorite neuroscience model right now because you have access to a lot of fancy tools. But the visual system in mice is very different from the visual system in primates. And it's really not clear at this point how much of higher level vision they share with us. So if you're interested, like me, in understanding higher level vision, mice are likely not a good animal model. So what's left? Well, what's left is the animal model that we have decided to study, which is the ferret. So this is the ferret. The ferret is a carnivore like the cat. And it turns out that carnivores have a visual system that is organized pretty similarly to that of primates. So there are nice and established similarities in, for example, how primary visual cortex, or V1, is organized in carnivores relative to that in primates. And so in that sense, carnivores, or in this case the ferret, is a good animal model for studying vision in general. But it turns out it's a really fantastic animal model for development for the following reason. Ferrets are born ridiculously immature. So basically what you see on the upper left is a baby ferret just a day after birth. At this time point, you can see they're only a couple centimeters long and they weigh around 10 grams. And their brain, as you can see down here, is teeny tiny, doesn't have sulci and gyra at that time point, almost looks like a rodent brain, like a mouse brain. And then over the next 30 days, all that they do essentially is grow massively. They open their eyes when they're 30 days old. So then that's roughly how they look like, and here's how their brain looks like. So you can see that there's a massive development in brain size. And again, they open their eyes at 30 days. So this is when visual development fully starts. And so you have a very long time period, both before their eyes open and structured visual experience starts, and then after that to access the brain and simply track as these processes develop. So that makes ferrets a really unique animal model for following development. In general, the usual standard neuroscience tools are available in ferrets that you can use in any other animal model as well. So in my own lab, what we're using is behavior in ferrets. We're recording from neurons using these extracellular probes. We're doing 2-photon calcium imaging, and we're doing anatomy as well. All of the data that I'm going to show you in the rest of the talk all comes from these physiology experiments. Okay. So with that, I will stop with the introduction. Does anybody have a question at this time point? All right, so then I will move on and tell you we'll move on from the introduction, and I'll tell you what this talk in particular will be about. So as I said, what I'm interested in is the development of higher level vision. And in this particular case, what we're going to look at is the development of one particular, more complicated motion function. And the particular function we're going to look at is called motion integration. And to give you a real sense of what I mean with that, let me show you this video, which hopefully plays. So I hope what all of you seeing is essentially a big flock of birds, or in this case, even 2 flocks of birds that are moving all together. And you can sometimes even take it further apart, but you still one group of birds moving in one direction in front of a second group of birds that is moving in a different direction. For you to be able to see that, what is happening is that you're pulling together the motion signals of these individual birds, and you're integrating all of those what we're going to call local motion signals into one big global signal, which is the motion of the entire flock. And so that's what I mean when I say in the rest of the talk, motion integration. In the ferret, and so this is a ferret brain, this kind of motion processing is handled by a pathway of cortical areas, very similar to what you would find in a primate, like in a human or in a monkey brain. And so that series of areas is usually called the cortical motion pathway. So as you probably all know, vision starts with the eye. From the eye, it is then sent to the thalamus, and from the thalamus to the back of the brain, in particular primary visual cortex, which I am calling V1. So V1 is the first cortical stage that processes visual information. Visual information from V1 then is sent back forward in the brain to a series of what we're going to call higher visual areas. So all of the areas that follow after V1. And one particular area that's relevant here is PSS, which is simply one of those higher visual areas. In terms of motion processing, what I just told you is that for motion integration, what you need are two things. You want to extract the local direction of individual elements. So think the birds, right? You need to encode all of these individual birds and which direction they're moving. And then in the second stage, you want to integrate those local directions into a global direction, meaning the movement of the entire flock. Another example of this kind would be looking at a tree that's blowing in the wind. You would want to encode the movement of the individual leaf and pool all of that together to encode the movement of the entire tree as it's moving with the wind. So these two stages are basically assigned to V1, which handles the local direction signals, and PSS, which handles the integration. Okay. Before we move on, let's make that even more explicit by talking about the functions that these two stages need to fulfill their role, just so that it's really clear as we go on for the rest of the talk. So V1, as I've said, is responsible for extracting the direction signals of local elements. So very fundamentally speaking, what it needs to be able to do that is it needs to be able to figure out in which direction something is moving. So the neurons in V1 need to be direction tuned. They need to be able to say something is moving left versus right or up versus down. PSS still needs direction tuning because it still needs to be able to say in which direction something is moving. Only the something is larger than what is represented in V1. So PSS needs to be able to say that entire flock is moving left versus right or up and down. So PSS still needs to be direction tuned. But then on top of that, it also needs to be able to integrate because that's how it computes where the flock is moving, by integrating over the local signals that it receives from V1. So two functions that are relevant for PSS, one is shared with V1. Okay. So you could then move on and make two very simple hypotheses for how this whole system is built during development. The first hypothesis likely will be something like this. You would assume that simple functions develop before complex functions. So basically, you would assume that direction tuning in both of these areas needs to show up before you can integrate. First, it's similar to like you first need to crawl before you can walk. You first need to be able to figure out in which direction something is moving, and then you can do more complicated math and complicated computations based on that. So this is sort of a prediction in terms of in which sequence functions develop from simple to complex. Your second hypothesis makes a very similar prediction, but it now looks at areas rather than functions. And it basically says, well, you probably need the lower areas, which are sort of at the bottom of the hierarchy and feed into all of these higher areas. So you need those lower areas to develop before the higher areas can figure out what their job is. And so what that would mean is that you would assume that V1 has to develop before PSS. And so here's how the rest of this talk is going to look like. I will walk you through the timeline of development for the direction selectivity in V1 and PSS. And then I will walk you through the timeline of how motion integration develops. And just in case you get lost and to organize your thoughts for the rest of the talk, I will tell you the take-home message of this talk already. And the take-home message is that what you will see from these two parts is that our first hypothesis is correct. Simple functions indeed do develop before complex functions, at least for the example of direction selectivity and motion integration. But what turns out to be very much a naive assumption is that the development of the areas is sequential, that V1 needs to finish developing before PSS can develop. Instead, what we find very consistently is both of these areas are developing in parallel, which is really interesting because what it means is that development is much more dynamic. Different stages of the pathways are set up basically in parallel and have opportunities to interact with each other to sort of guide their development back and forth as is in a loop. And it makes studying it a lot more complicated because it means that we have to look at all of these stages at the same time point to figure out how development happens. But it's a really interesting way, and it's a really powerful way in which our visual system is shaped. It's much more complicated for sure than the simple assumption that one area first, then the next area after that. Okay, so let's talk about direction selectivity and how that develops. When we measure direction selectivity in the lab in these experiments, we usually use stimuli that look like these. These are called gratings. And we usually have the gratings, at least when we measure direction selectivity, move. And they usually move orthogonal to their orientation. So this particular grating could move either left or it could move right. And then we call a neuron direction selective if it really strongly responds to one direction and doesn't care at all about the other direction. So a neuron that really strongly responds to this thing moving left and doesn't respond at all to it moving right would obviously be direction selective. So here is some real data, and I should say all of the stuff that you're seeing today is the work of a super talented graduate student, Augusto, who was originally from Argentina, who is now a postdoc at the Max Planck Institute in Florida. So here's what Augusto did. Basically, the curve on the left shows you an example direction tuning curve for one neuron in PSS. What this means is that on the y-axis, we're plotting how much the neuron is responding to different gratings, moving in different directions. And along the x-axis, what we're plotting is the direction that the gratings are moving in. And we've organized this plot in a way that 0 corresponds to the direction that the neurons like a lot. This neuron likes a lot. And you can see that you get a really nice, strong response to this particular stimulus. And then correspondingly at 180 and minus 180, this is the opposite direction. So if this was up, that would be down. And you can see that this particular neuron does not respond at all to gratings that are moving in this opposite direction. And so we would say that this is a very direction selective neuron. And we can quantify that by computing what we're going to call a direction selectivity index. And so basically this index compares the response to the preferred direction to the response to the opposite direction. And it takes a value of 1 for neurons that are as strongly direction selective as this one. So they respond only to the preferred and not at all to the opposite direction. And if this index is 0, it means that the response to the preferred direction is as strong as the response to the opposite direction. So that neuron has no idea whether something is moving up or down or left and right, because it's responding exactly the same to both. Okay, so this is one PSS neuron. We obviously want to record this for many neurons at once, not just in PSS, but in V1. And all of this now is in adult animals. And so when we do that, what we get is a distribution of direction selectivity indices for the two areas. And I'm plotting those two distributions here. And I'm plotting them in the form of a cumulative histogram, which many of you might have seen already, but I know these tend to be confusing. But since I will be using them throughout the rest of the talk, let me just very briefly explain to you how to read them, just as a reminder. So in all of these, what we're plotting along the x-axis is one particular parameter. So here, this happens to be the direction selectivity index, which ranges from 0 to 1. And then on the y-axis, we are plotting the cumulative count of neurons. And so the way to read this is, for example, to look at the value of 0.5 and then find the line up here and look at the corresponding y value. And what that tells you is how many neurons in our overall sample had direction selectivity indices of 0.5 or lower. So it's almost like you're plotting a histogram, but then you're summing across all of the bins until you hit 0.5. Okay, the most important thing that you get from this is the observation that if you're looking at the direction selectivity index, values tend to be lower in V1 than in PSS. And again, this is in adult animals, which isn't entirely surprising. What it basically tells us is that PSS is more specialized for motion processing because it's more selective for it than V1. And again, V1 is sort of at the bottom of the visual hierarchy. It feeds to a lot of different visual areas. So it's not that surprising that it's not yet as specialized as an area like PSS, which is sort of further down the hierarchy and likely to be more specialized for whatever function it's carrying out. And because we're interested in motion, we've picked an area that plays a role, as I've said, is part of the motion pathway. So not a big surprise here. But it's still important to see that there is a difference. Okay, what we can ask now that we've done these experiments in the adult is, how does that develop over time? And here's what we find. So as I've said, ferrets open their eyes when they're about 30 days old. And so this first group of animals are all animals that have barely opened their eyes, if at all. So they have barely any sort of good visual experience, clear visual experience, not just blurry through the eyelids. And so that data will be shown in light blue. And then the next group of animals that we investigated are about a week older. That's in dark blue. And then the adults are all in black. And so again, the cumulative distribution for the direction selectivity index is shown here for V1. And here's what you can see. For the young animals that barely have their eyes open, the direction selectivity is really undeveloped. The indices are really low, meaning these neurons are not really direction tuned at all. But then within a week after eye opening, from the light blue to the dark blue, direction selectivity drastically develops, actually to the level that you see in adults, if not better. And then it doesn't improve that much further. If at all, it might get a little worse. So basically, direction selectivity develops in the first week after eye opening in V1. Okay. It's a fact at this point. Maybe not that interesting, but it does become interesting when we look at when PSS develops. So here is the same groups of animals, but now we're recording in PSS. Same basic observation. If we record just at eye opening, direction selectivity is much less developed than in adults, which is the black line. And it develops very strongly in the first week after eye opening. So the same jump from the light blue to the dark blue happens in PSS as well. If my prediction had been correct that V1 develops before PSS, what I would have expected is that this timeline maybe wouldn't have made it all the way over yet. It maybe might have made it like halfway, because it would have needed V1 to be completely done first before it could have developed fully. But that's very much not what we find. I still don't want to rule it out entirely, that PSS develops after V1, because obviously we have very coarsely sampled at this time point. But as I will show you in the next couple of slides, for most of the things that we have looked at, it seems like V1 and PSS are really going in parallel. And in some cases, which is what I'm going to talk about next, it almost seems as if PSS is actually going in front of V1, the opposite of what I had predicted. Okay, hold that thought in mind. I just very briefly wanted to take you on a very short detour since we have this data up. One of the things that we noticed that we still have to sort out more is that consistently throughout development, PSS is better than V1 at direction selectivity. Remember, I said that was the case in adult, but these plots show you it already is the case very early in development, even in these animals just around eye opening that have very little direction selectivity yet. And that might tell us something about the circuitry that links V1 and PSS. So if PSS is able to get better at direction selectivity because it has some machinery in place that allows us to sort of improve on the signals it's getting from V1. So then it seems, so likely the feed-forward circuits that link V1 to PSS, it seems like that machinery is already in place when the animals aren't even ready fully to compute this thing yet. So basically, even though the areas themselves are not that good at computing direction selectivity, whatever makes direction selectivity in PSS be better than V1 exists from the get-go, which is also really interesting because you could have assumed that as part of the sort of sequential development, PSS would be much more similar to V1 in the get-go, and then eventually figure out, oh, I'm supposed to be a motion area, so I better get better at figuring out how direction works. But that seems not to be the case. It seems to know how to make direction selectivity be better than in V1, basically from the start. Okay, so let's pivot back to the main thrust of what I wanted to tell you about. So what I'm left here at this time point is that V1 and PSS seem to be developing in parallel. And I promised you I would show you an experiment that pulls that even more into question, and it shows you that PSS might be actually going before V1, sort of putting the hierarchy on its head. So here's how we can say that. This is using a pretty cool paradigm. Because, and I say cool because it allows us to watch development happen online as we're doing experiments. And it works as follows. In these experiments, we take ferrets that basically don't have their eyes open yet. So as you've just seen, they have immature levels of direction selectivity in V1 and PSS. And we anesthetize these animals, and we park them in front of a computer screen so we can show them stimuli, and then we open their eyes. And so we basically, for the next 10 hours, these are sort of annoyingly long experiments. For the next 10 hours, what we're doing is we're showing them particular visual stimuli. And throughout that entire time period, we're recording activity both in V1 and PSS at the same time. And so basically, what this allows us to do is to watch what the areas are doing as the animal is gaining visual experience with something that is entirely under our control. And you might question why this could work in the first place. It seems 10 hours in a ferret's life should be rather short compared to sort of the overall lifespan of a ferret, or the development, which I've shown you might take up to a week. But it turns out baby ferrets sleep an awful lot. And so probably having your eyes open for 10 hours and showing it and looking at a stimulus is probably actually a couple of days worth of life in a baby ferret, for a baby ferret. So it's long for the graduate student in the experiment, and it might similarly be long for the ferret in terms of looking at the stimulus. So again, the experiment is recording in PSS in V1 while we show the animal a particular kind of stimulus for a very long time. And throughout that, watching what's happened. So here's what we find in these experiments. First off, this is the distribution of direction selectivity indices in PSS before we do anything. So this is the same data you've just seen, immature levels of direction selectivity. So now in this first set of experiments, what we're doing is, as we have the animal parked in front of the screen, we're showing it a drifting grating. And the grating, we are constraining to drift along one axis. So we're not really probing directions. We're not really training direction selectivity, actually. All that we're doing is we're showing a grating that's drifting to the right, or we're showing a grating that's drifting to the left. And then because we don't want to blast the visual system entirely, we're interspersing that with gray screens. Okay, if we do that, if we show a drifting grating for 10 hours, what we get is the following. A direction selectivity index curve that has shifted to the right. So what that means is the neurons have become more direction selective in these 10 hours, which is why I've been saying we can watch development online as it's happening. And the cool thing is, it turns out that that is actually because we have been showing the ferret a drifting grating. The same thing does not happen if you show the ferret a static grating. So the black line is the same 10-hour period, but we're showing a grating. We're just not making it move. And you can now see that the curve is not different from what we had at the start. So what that tells us, and this is sort of a side point to my story of the sequential development, but it's an important point. What that tells us is that visual experience might be important for driving the development of direction selectivity in PSS, and that the kinds of visual experience matter, that it's not just sufficient to show any kind of visual stimulus, but that we actually have to show a drifting grading or something that moves if we want direction selectivity to develop. Okay, but if we go back to the relative development of the areas, here's the more relevant time point. The same thing happens in V1. I don't have the static data for V1, but you see the same thing. You initially have an immature distribution of direction selectivity that improves after 10 hours of showing a drifting gradient. Okay, fine. So the major point that I wanted to make is that we can use this paradigm to watch as the direction selectivity shows up. And so that allows us to now ask, sort of with much more precision, who goes first? And here's what we're currently finding. So basically what we're doing now is we're plotting the data at the start of the experiment, at the end of the experiment. So these are actually 7 hours rather than 10 hours. And this is the shift that I've just talked about. And then we're plotting them at the halfway point after 3.5 hours. The top plot is for V1 and the bottom plot is for PSS. And what you should see is that the shift that the data makes after this halfway point, after 3.5 hours, is actually smaller in V1 than it is in PSS. So PSS has made a larger jump, has developed more in this time period than V1 has. And we don't just see that if we combine all of the animals into one big group. We can even see that when we look at individual animals. So this is all of the animals that we've used in these studies. What we're plotting is every circle is one particular animal, and we're plotting the median direction selectivity index for that animal at the start of the training, the halfway point, and the end of the training. And so what you should be able to see is that in V1, really the major improvement is towards the end of the time period. Versus in PSS, you already can see that all of these dots have moved up after 3.5 hours. So what this shows you is that I haven't gotten to the point about simple functions before complex functions yet, but what it shows you is that development very much is happening in parallel in both V1 and PSS, and may even be sort of backwards, and that the higher order area, which you would have assumed depends on what the lower area is doing, is actually developing before the lower area. And so maybe actually in development, visual information sort of flows backwards, in that the higher order area first figures out things and then informs the lower area in what it needs to do. And you'll see that in the next part, there's sort of a similar story along the same lines. One of the things that we're interested in doing now in these kinds of experiments is trying to look at dependencies, whether both areas are actually necessary for direction selectivity to develop, or whether we can actually inactivate one of the two to figure out who really is important for this to happen. Okay, so this was, as we have said, the sequence was simple functions first, complex functions after. So now let's finally talk about complex functions. When I introduced the concept of motion integration, what I talked about was a flock of birds and how you can see that as one entire cloud moving together, or the tree was the other analogy. In the laboratory, we need a somewhat more controlled situation to probe motion integration. And the default stimulus that everybody has been using for this purpose has become these stimuli that we're calling plaids. And so they look, if I can get this to work, they look like this. So I hope what all of you have seen is something that is jointly, nicely moving downwards, right? It turns out that in reality, what was on the screen were two gratings that simply occupied the same region of space. One moving over and to the left, and the other one moving down and to the right. But because of the way that they were superimposed on top of each other, what you perceive is something that is jointly moving downwards. You are not able to tease these apart in the situation that I've just set up. You simply perceive one pattern that's coherently moving in one direction only. And so introduce some nomenclature. What we're going to do is we're going to call these individual gratings the components, and they have accordingly their component direction. And then we are going to call the integrated thing the pattern with its pattern direction. Okay, so what this allows us to do now is to probe how neurons are responding to these patterns. And then we can ask them the following questions. We can ask each neuron whether it sees one direction in these plaid stimuli, the same way that you saw only one direction perceptually when you looked at them. So if the neurons are seeing only one direction, the only way that that can have happened is because they've integrated the two component signals that were present. And so that's a signature of neurons that are able to integrate if they see only one direction in these plaids. And we're going to call those neurons the pattern neurons. On the other hand, neurons may not be able to integrate. And even though that's impossible for you to do perceptually, they will see the two components moving along their directions. So they will treat the plaid as if it actually has two motion directions present that correspond to the two components. And so that's a signature for neurons that do not integrate, that they see two directions in the plaids, and we're going to call those neurons the component neurons. Okay, with a lot of different math and things that we're going to skip over entirely, we can basically do that for a lot of neurons in both PSS and V1. And here's what we find. So when we summarize how neurons are treating these plaids, we're computing two numbers for every neurons, and then we're plotting them in these kinds of plots that I will very briefly walk you through. So along the y-axis in these plots, we're plotting a number that tells us how pattern-like a neuron is. So the higher this number, the more the neuron looks like a pattern neuron. And then along the x-axis, we're plotting how component-like the neuron is. So how much, this basically tells you how much are they able to integrate, and this tells you how much they're not able to integrate. And then we usually divide, and so every dot in this plot now is a neuron. And we generally divide these plots into two regions, or three regions, but the two important ones are this upper part that we're calling the pattern neurons. So this contains all of the neurons that can integrate. And then we're taking this lower part as the component neurons, which are the neurons that very clearly do not integrate. Okay, so here's how, if you record from a bunch of neurons in PSS, here's how they fall into this plot. And the really important thing to see here is that there are neurons in this upper part. So PSS contains neurons that integrate. And it turns out there's a similar area in primates, and both monkeys and humans, where you have neurons that integrate the same way. So that's why we consider PSS a higher order motion area that can handle motion integration, because it has a proportion of neurons that show the signatures of integrating local motion directions. And again, it's because they see the plaids as moving in one direction. And we're even more confident in that because we can find that in PSS, but we cannot find it in V1. So this is not something that PSS inherits from at least from V1. It's something that comes up later. Because if you look in V1, all of the neurons are down here in the parts of the neurons that don't integrate. Okay. And then a last way in which we can look at this difference and degree to which an area can integrate is by computing something that's called the pattern index. So this deals with the fact that this assignment of neurons into different categories is a bit arbitrary. As good scientists, what you should have seen is, or what you should have argued with me is that this looks like one big clouds of dots, and it's really arbitrary how I've drawn different lines through it and have assigned names to the neurons. And so the pattern index deals with the fact that this is so arbitrary. And essentially what the pattern index is the difference between these two numbers that I'm using to characterize the neurons. And so it subtracts the thing that tells you how pattern-like the response is from the thing that tells you how component-like it is, or actually the other way around. So positive numbers now with this pattern index are neurons that integrate, and negative numbers are neurons that don't integrate. And again, this is a cumulative distribution, but the important thing is that the PSS line is shifted to the right of the V1 line, meaning there's more integration in PSS than V1. Okay, so if you were, I hope you're not too lost, but the idea is that in PSS, there are signatures for motion integration that we get from looking at these PLAD stimuli. And all of this was in adult animals. So when does this show up in development? And here is our answer. So again, we're recording in animals of different ages. We started with a time point in which direction selectivity was already developed, because in the end, to be able to do this kind of analysis, you need a basic level of tuning. So the blue line is the last point of the data that I've just shown you on the direction selectivity. These neurons are direction selective at this time point. And then progressively, the red line is a week older and the black is in adults. And the main takeaway, to make sure that we don't run way over time, is that basically we see that motion integration develops basically a week after direction selectivity develop. You get to mature levels in these animals that are around P44, which is a week after direction selectivity has developed. So in that sense, it's very much true that you get direction selectivity first, motion integration after. The really cool thing that goes back to these interactions between the areas is that more or less by chance, we also were recording in V1 during the same time period. And I've just told you that V1 basically only has neurons, or mostly has neurons, that don't integrate. And that was important because it basically is the specialization that shows up in higher level cortex. So the interesting discovery that we made is that is the case in adults. So you can see, again, most of the dots, each of which corresponds to neuron, are in the lower right. Same is the case in these animals that just have developed direction selectivity, but there's a phase in between where in V1, you now get pattern cells. This is a temporary thing. So it starts out mostly component, becomes more integrative, not at the level of PSS, but nonetheless, it has more of the signal and it goes back. And so we're still puzzled by this. What we do know is if we remove PSS by inactivating it, that goes away. And the responses become entirely like they are in the adult. So there's something that PSS is telling V1 about how it wants signals, what kinds of signals it wants during development. At least that's what we think, that makes this effect show up. So it's maybe something in terms of the kinds of signals that PSS needs to properly develop that pulls V1 off course temporarily, for example. But that's one of the things that we're actively investigating going forward. And so this is just the active inactivation data, which we'll skip over. So again, to get back to my take-home message, I've shown you that, yes, indeed, it seems simple functions in the visual system develop before complex functions. But very much what we're seeing is that development is a very complicated event, which you probably would have expected, that happens at many, many stages of the visual system in parallel, with a lot of back and forth across these stages. And that this back and forth is probably very important in setting up the visual system correctly. And I wanted to end with this picture, because what often happens if you listen to any of us talk about the visual system, about the brain in general, right? What we will tell you is that there's all of these different areas and that we can somehow organize them in some kind of hierarchy. So this is shown for the human brain with some of its connectivity on the left. The right is a monkey brain with the visual areas highlighted and all of them organized in the spaghetti-like diagram in terms of hierarchy. And so we know that there is these hierarchies, that areas are densely interconnected, that the whole thing is a network, that information flows forward and back rapidly and goes in loops. But then really what we often do when we study the brain is we go one area at a time and sort of treat it in isolation. And we then will shrug our shoulders and say, well, yeah, we know it doesn't exist in isolation and it obviously gets information, it sends information, but we still study it in isolation. And I think when it comes to development, that is really sort of a dead end, because I think we we are likely to massively misinterpret our results if we don't look at what is happening in other areas at the same time that might explain whatever we're finding in the area that we're currently studying. Case in point, this change in the V1 tuning properties that I just talked about. Okay, not to keep you too much longer, let me think. Basically, all of the people in my lab, as I've mentioned, Augusto is the person who's done all of the experiments. And because I know that part of this is to tell you about the graduate program at Hopkins, one of the things that for me sort of stands out about our graduate program and Hopkins in general is how interactive and how collaborative the environment is. And so here is basically my lab in a recent Zoom lab party that we threw for Erica, who was graduating. We were playing a murder mystery game. And I'm showing you this not just to give credit to all of these fabulous people, but also because it shows you that basically I have merged my lab with the lab of Ed Connor, who is at the Mind Brain Institute as well, who is similar to my lab, interested in higher level vision. And so all of our big lab parties and everything, it's as well as the lab meetings, we basically have together and we function as one big lab. And I don't know many places other than Hopkins in which that would be so easily possible. And so Ed and I work a lot together, but Thomas, for example, now is a graduate student who's jointly supervised by me and Dan O'Connor at the same time, because we've started to become interested in somatosensation in ferrets. And so Chris will confirm this, as will the students. There are many, many cases in which students are working across multiple labs. Okay, and with that, I will stop.
Thank you, Kristina, for a wonderful talk. And I have to tell you that I've seen vision research talks in the past, and yours, I understood every single part of it. So, because when we go into the biophysics, I get a little lost, I have to say. But I understood, and it was a wonderful experience. So we have some questions for you. First, from Dr. Garcia Araras, he says, does the velocity of the motion has an effect on the development of the VI or the PSS?
That's a very good question. So it probably does, but it's interesting in it that it seems like it may be less acceptable, it may be less accessible, sorry, is what I mean, to shaping. So in this experiment that I talked about, where we basically watch development online, it turns out you can make neurons be direction selective, but you can't, if you show things of a particular speed, the other ones that have other speeds still develop, versus if you show things of a particular direction, only the ones that have the matching direction, or mostly the ones that have the matching direction, develop. So we think we can, like, you can maybe imprint direction a bit more on the developing system in terms of which sort of preferences will show up. But with the speed, it seems like you get all of the different speeds independent of what you have shown. But that's something that, yeah, is to be determined.
Okay, thank you. And then there's another question from one of our undergraduate students, Cielo. She says, can ferrets serve as an in vivo animal model to study the effects of artificial light pollution exposure on brain development?
Again, to study the effects of
artificial light or light pollution exposure on brain development.
Yeah, yes, yeah. So ferrets are, the big advantage of ferrets is that you have basically access to a lot of development that otherwise would happen in utero, and that you have a really long period in which they don't have their eyes open yet, and things are developing, and so you can actually, because they're no longer in utero, you actually determine how they're growing up. And so you can raise them in the dark, you could raise them in a very bright environment. There's all sorts of things that you could do.
Okay, thank you. So any other questions that you can unmute yourself and ask Dr. Nielsen before we head on to the recruitment part? I'm shy.
I'm not going to bite anybody.
see it. Okay, well, thank you, Kristina. We appreciate your. So now we can move on to the recruitment component. Of course, we'll have Dr. Nielsen and Dr. Porter and some graduate students from Hopkins that have joined the call. So, Chris?
Yeah, I'll take it away. Sure. Let me present my screen. Let's see. Do that. Can you see that?
Yeah, yes.
Great. Let me do full.
Yep.
Right, so this is kind of an overview of the program. I hope to give you a flavor of what it's like to be a graduate student in our program. So these are some of the key players in the neuroscience training program. I'm a co-director along with Jim Knierim, who's here on the side. Can you see my pointer when I'm pointing?
Yes.
Okay. And the other folks that you might get to know, Angelica Dutzelhofer, she is the director of admissions. So she's the one who's in charge of running the admissions every year. And Rick Huganier is the director of the Department of Neuroscience, which the neuroscience training program is under that. Just some overview. So admissions, the deadline for your application was December 1st, so you can't apply this year, but I'm hoping people in the audience are going to be applying for next year if they didn't already apply for this year. Recruitment 2021, if you did apply, we're going to have 2 recruitment events, one January 25th to 26th, and the other February 7th to 8th. And we typically recruit about 16 new students every year to the program. In terms of funding, all students are guaranteed support throughout your PhD, so you don't have to worry about that. If your PI doesn't get his R01 grant renewed, the program will cover you. But they'll get their grant reviewed, don't worry. They'll also get a full tuition waiver, and you get a stipend. So you get about this for 2020 to 2021 next year. The stipend is just over $34,000. It's not a huge amount of money, but it is enough that you can live actually very comfortably in Baltimore. You also will have medical and dental insurance covered. In the program right now, we have 85 students enrolled, 83 PhD candidates and 2 MD PhD candidates. And the time to degree, how long it takes to go from beginning when you first start to the very end when you submit your thesis and defended it, is about 6 years. We're trying to get that number to about 5.5 years. So the neuroscience faculty, so we have 109 total faculty that are part of the neuroscience program. 31 of those have their primary appointment in the Department of Neuroscience, like me and Kristina. Which is part of the whole program faculty. There are 19 different departments that are part of their neuroscience program and 7 different institutes. So shown here are just some of those examples. So most of the faculty are at the Hopkins School of Medicine. That's where I'm located. And a lot of different departments are in the School of Medicine, including the neuroscience department. A large number of faculty are also at the Krieger School of Arts and Sciences. That's actually where Kristina is located. She's at the Mind Brain Institute. And we also have some faculty that are part of the Department of Biology, which is separate. They're part of this, the School of Arts and Sciences. There's a number of other institutes. So the Kavli Institute, this is an institute that started a couple years ago now, and it's a collection of scientists, neuroscientists, engineers, all towards neuroscience type of adventures. The Lieber Institute is located in the same building that I have my lab in. They are interested in neuropsychiatric diseases and doing large-scale sequencing efforts of different genomes to try to figure out the basis for different neuropsychiatric diseases. We also have faculty at the Bloomberg School of Health, the Brain Science Institute, and lots of other different places. And I'll talk a little bit more about HHMI in a second. If there's any questions, I can't see anybody on my screen, but if there's any questions, please feel to interrupt. So this is to give you an idea of what it means to have that many faculty. So this is just some of the pictures. This is not even all of the faculty that are in the neuroscience program. And this is really just to convey to you what it means to have this many faculty. It just means choices. You'll have a large number of choices when it comes to figuring out where you want to do your thesis work. And chances are that whatever you're interested in, there's likely a faculty member, there's somebody in the neuroscience program that's working on that particular aspect. We also have the opportunity to do your thesis work at Janelia. This is part of the HHMI, the Howard Hughes Medical Institute. They have a campus at Janelia, which is in Leesburg, Virginia. This is about an hour and a half drive from Baltimore. And they do neuroscience research over there. And a lot of times the work that they do is essentially funded by HHMI, which means they can do things at a grander level than most regular research labs can do. And it's we have a unique arrangement with them that you can be a neuroscience graduate student at Hopkins, but then do your thesis work at an investigator over there. What we also have set up is that, and this is an agreement we have with Janelia, they have essentially it's like a hotel on their campus. And so when you begin as a graduate student, so this will be for August 2021 for this coming year, we have what we call a boot camp. And what the boot camp is essentially designed to, the real reason is to get all the first years together in the same location, and you get a chance to chat with your compatriots, you get to talk to them, you get to know them, because you'll all be staying on site together. They have a bar there, they have a restaurant there, you can really get to know the students you're going to be spending a lot of time with. And also you'll learn some neuroscience, and you also do some computation. So to get your feet wet, and how do you do MATLAB, and how do you do those types of data analysis. But really what we set it up for was just to get the first years together, to start bonding together. So class curriculum. So most of your classes will be done in your first couple of years. There are some courses that go over many years, and that's like Journal Club, for example, which is one of the courses that Kristina runs. The main neuroscience course is called Neuroscience and Cognition 1. We just abbreviate it as NeuroCog 1 and NeuroCog 2. It's a full year course. NeuroCog 1 is the first half year, and NeuroCog 2 is the second half of the year. And this course pretty much goes over everything you've ever wanted to know about neuroscience. It's the fundamentals of neuroscience. And we set this course up, with the knowledge that not everyone comes in with the same background, and that is something we prefer, that everyone's coming to this a different background and different experiences. And you might not have been exposed to molecular neuroscience, or you might not have been exposed to systems neuroscience, and that's totally fine. And this is what this course is really designed to, is to get everybody up to the same page, and it'll cover all those different topics. We also have a number of other required courses. We require everyone to take a statistics course, unless you can if you've already taken one before, you can err out of that. We also require quantitative methods course, because quantitative methods are very important in a lot of modern neuroscience. A circuits and brains disorders course, where you learn about neurological diseases. Journal Club course, this runs, you're required to do this for your first to third years. We do have a teaching requirement. The teaching requirement is not that, it's not that much, to be honest, but it can be depending on what your interests are. So if you're interested in teaching, you can arrange to have a more, long-term teaching. But most of the time, you're just TAing the NeuroCog 1 or 2 course. We have a seminar series. It's a really fantastic seminar series. We typically get about 30 speakers every year. Lunch is provided. This will be in when we get back to normal. And there are 2 graduate student invited speakers every year. The lunches are great because you get a chance to talk to the speaker and find out essentially, whatever you want really, but a lot of times the students use it as an opportunity to figure out how that particular speaker got to where they are, what are some challenges, just to get to know them, get to know the back end of doing science. We have a career skills course for 3rd and 4th years, and this is to expose you to all the different types of careers that you can do with your PhD. We understand that not everybody wants to go onto academia, and that is also perfectly fine. And so this will also expose you to lots of other careers, such as policy or data science or whatever the case might be. We bring in people to talk to the students about that and to just expose you to lots of different opportunities that you'll have. And we also have, starting next year, a grant writing course and so a paper writing course. We have a requirement for 3 electives. There are over 100 different options you can choose from. We have 18 that are offered by the neuroscience program itself. These include courses like Writing About the Brain from David Linden. So David has written a number of books. I think his last book came out in August of this year. So you can learn from someone who's actually doing this. You can learn how do you write about the brain. You can take like a psychedelics course from Gould Dolan, or if you're more interested in cognitive neuroscience, Jeremiah Cohen has a course, and so forth. So there is tons of electives that you can take, and they're really catered to what your interests are to help your research go forward. So as Kristina mentioned, the Hopkins is, I think, a fairly unique place of all the places that I've ever been, and it's highly collaborative, and it's hard to convey what that means unless you're actually here, but I'm going to try my best. And so let's say, for example, this is David Linden. So let's say you're in David Linden's lab, and you can see who is David Linden collaborated with just in the Department of Neuroscience. And you can see that there's a number of different faculty that he's collaborated with. So Saul Snyder, Randy Reed, Rick Huganier, Paul Worley, Valina Dawson. So these are all just labs. If you look at his publication record, you'll see that he's collaborated with. And you can take any of these faculty and see who they collaborated with in the Hopkins Neuroscience program. So if we take Paul, for example, he's collaborated with Jay Baraban, Hei-Kung Lee, Kerry Martinovich, Shan, so it goes on and on. And this is true of all the faculty. And it's a really wonderful place to do research. So what does this really mean as a graduate student? It means that you won't get stuck. Whatever you need to do in your research, there is going to be a faculty member, someone in the neuroscience program that's willing to help you. If you need someone to show you how to do the Blender program, I'm sure Kristina's lab could help you with that. Or if you need someone to help you with electrophysiology or RNA-seq or whatever the case might be, there's somebody in the program that is going to be very happy to help you with that. So as a graduate student, it means that you can always move your research forward. You won't get stuck. So mentoring is also super important to us. And so this is just to show you the types of mentoring that you'll get as a new student. So here you are as a Baby Yoda first-year student. You just started. So the first thing that we set up are a big SIB. So for example, you maybe get John. This is usually a senior graduate student in the program, will be your big SIB. And this will act as someone who you can talk to before you even get here about, where should I live? What are places that, where are good restaurants to go to? What's life going to be like when I get to Baltimore? We also have a pre-thesis advisor. So this is a faculty member who can also address questions like, what courses should I take? How many courses should I take? Those types of questions you might have. By around your second year, you'll join the lab, so you'll have a thesis advisor. So for example, you join David's lab, and you'll set up a thesis committee. So you'll have 3 additional faculty on your thesis committee. And I think the dirty secret is that the thesis advisory committee is meant to be on your side. It's not necessarily meant to be on the thesis advisor's side. The thesis committee is really to support you and to make sure that you are progressing in your studies, and that if any conflicts that arise if your advisor for whatever reason says that you have to do one more experiment, your thesis committee members are there to support you and say, look, this person's done, it's time for them to graduate. So just keep that in mind. That's what they're meant for. And on top of all this, we also have the steering committee. So there are 12 other faculty in the neuroscience program that are overlooking all the students in the program and making sure all the students are doing well, that all requirements are being met, and that nobody is struggling. And if somebody is struggling, to get them help. So all these different people are on your side throughout your entire career here at Hopkins. On top of that, we have something relatively new that was started in 2019, and this was set up by the Committee on Diversity and Inclusion. And these are what we call graduate life advisors. And so these faculty are just an additional mentors that you can go to, and including Kristina, that can help you with other questions, things that you might not want to go to your thesis advisor about, more like work-life balance type of questions. If you just need someone to talk to, these faculty will be there for you for that. There are also a number of other programs at Hopkins for health and wellness. I'm not going to go over those right now, The website here is wellness.jhu.edu, so please take a look at that. They will go over all the different resources for physical health, emotional health, mental health, social health, etc. So Hopkins has a large number of support networks for you for that. There are a number of community events, so department events. So we have a neuroscience retreat every year. So this is one of those retreats. And we also have happy hour every week or every month now. So this is some students playing a rigorous game of ping pong. It's a chance really just to hang out and catch up with everybody. We also have a softball team. Our 2019 record, we have 2 wins and 16 losses, and those 2 wins were by forfeit. So I think we're definitely recruiting for some players. We also have a soccer team, the goal-directed behaviors. The 2019 record, we're doing pretty well. They had 1 win, legitimate, 14 losses and 1 tie. But again, it's not really about winning. It's more just about having fun with your fellow classmates. So where do students live? So this is a map of campus. And so the Hopkins is split between 2 main campuses. So there's the Homewood campus. This is actually where Kristina works. And there's the School of Medicine campus. And this is, for example, where I work. And there is a shuttle, which is what this blue line is showing. There's a shuttle that goes from one campus to the other. And this is a really great shuttle. It runs all the time. So it's a really great way to get to your different campuses. And as you might expect, a lot of the students actually live on the shuttle line. So these are just some examples of the students and roughly where they live on this map. So for example, here's Jessie. She lives in the Hampton area. She's standing in front of a row house that she just bought. So the stipend is enough that you can actually afford to buy a house as opposed to just renting. There's lots of other chances. So there's some other row houses. So William lives in one of these row houses. Alan lives in a little fancier looking apartment complex. Lionel lives in a row house. But they're all different neighborhoods. And that's something that's really quite special about Baltimore, is that it's a city of neighborhoods. And each of these neighborhoods have their own different flavor, their different style. So if you want more eclectic, there's neighborhoods for that. If you want to live more like a downtown where you can just walk to everything, there's neighborhoods for that. So there's something for everyone. And really, you should just talk to the grad students about this. They will give you all the information you need about what type of neighborhood and where to live and what it's like to live in these different places. There's lots to do in the city. Baltimore has lots of things going on. So for example, there's a farmer's market every weekend to pick up new fresh groceries. There's the Baltimore Symphony Orchestra. There's season passes for students, $35. There is the Hippodrome, where you can watch theaters and musicals. I think in the summer 2021, Hamilton is coming, so I hope that it's still gonna go on. The Baltimore Museum of Art is fantastic, and it's free, and it's right by the Homewood campus. Visionary Art Museum is another museum. It's quite eclectic and definitely worth checking out. It's right next to campus. There are places, there's lots of great food and drink. We have a football team, and we also have a baseball team. Things to do nearby. So there's lots of also things to do nearby Baltimore. So here are some of our graduate students who went on a hike in West Maryland. There's some beautiful locations for hiking. You can also go to DC. So DC is about an hour away. You can take a train. So there is the Penn Station, Baltimore Penn Station train station. There's a train that goes straight into DC, and it is $8 one way, so $16 round trip, and you can go to DC for the day. Outreach. So outreach is greatly important to our students, and there are a lot of different opportunities for outreach that the grad students are involved in. So these are just a couple of examples here. So one of the main ones is Project Bridge, and this was something that was set up by neuroscience students and is really expanded to a lot of other graduate programs as well. And so this is a chance for outreach into the community. So here are 2 of our graduate students who are showing this person, essentially how like electrical activity on your muscles, you can actually visualize that directly. And it's a really great way to go into the community and to really share your love of science with the community. Thread is also another type of outreach. And so this is where you can mentor students that might be struggling academically and who really just need to have someone to just help them with that and to show them, what a scientist is like, and that we're not scary, and that we can actually help them through these types of things. And you can really make a big difference in someone's life. And here is our website. So there is a ton of information on our website, far more than I can give you right now. So please take a look at that. If you just did a Google search for outreach at Hopkins Neuroscience, you'll find this website, and the URL is here on the top. And shown here are the different types of outreach initiatives that we have set up at the Hopkins Neuroscience. So Project Bridge and all those other ones are there. So please check that out. Diversity inclusion is also really important to us. So this is our website for that. If you do a Google search for that, you'll find it. And this is our URL here at the top. So with the initiatives we have set up for diversity inclusion are that the neuroscience department has a committee on diversity and inclusion. And this is a committee that's composed of faculty, postdocs, and students. And this is to organize different events that are on departmental wide. The neuroscience graduate students also have their own committee, the Student Diversity Committee. This is actually the largest committee we have for the neuroscience training program. It's very popular, and it also does involve in a lot of these outreach programs and also organizing events on campus. And just shown here, which is on this website, are all the different types of diversity initiatives that we have. The one I really wanted to talk to you about now is the summer internship program, which is, you can find more information on the website about that, the SIP. We have 2 students. So in summer of 2020, we had a student from UPR was there. She was in Jeremiah Cohen's lab. And in 2019, we had a student from UPR that was in Solange Brown's lab. This is Solange right here. So let me just talk to you a little bit more about that, because I think it's, the students in the audience, I think this is a really fantastic program to apply for. So the summer internship program. So we have 2 that are fairly related that the neuroscience department is involved in, neuroscience program is involved in. There's the Department of Neuroscience summer internship program, and then there's the Kavli Institute Neuroscience in summer internship program, and they're related. One is essentially funded by the neuroscience program, and the other one is funded by the Kavli Neuroscience Institute. For the Department of Neuroscience one, you will be hosted by a neuroscience department lab. For the Kavli one, you'll be hosted by a Kavli Institute lab member. So the Kavli has some neuroscience members in it, but they also have engineers and other related areas. So they're somewhat overlapping, but there are different types of focus in the Kavli Institute. And you can just look this up on the website to see which one you would like. Some of the details: it's a 10-week summer program. It provides you hands-on research experience. You will get a stipend and housing at Hopkins. This will all be included. This is something I wanted to stress, that it's eligible for any college year. You don't have to be a junior to apply for this. This is incorrectly stated on the website. So I just wanted to point that out here, that you can be a freshman and you can apply for this. You can be a sophomore and apply for this. It's open to any college year. It also includes summer lunch series. So this is a chance where it brings in different faculty and other students together to talk about how do you navigate careers in science, how do you navigate academia as a URM, how do you apply for graduate school, how do you apply for fellowships, and you also get you more mentoring from other faculty. When you apply for this program, and the link is on our website, and you can also Google search it, when you apply for it, you'll be applying for both at the same time, unless you say otherwise. Otherwise, you're essentially, with the same application, you can apply for both. The application deadline is February 1st, so it's coming up, and this will be for the summer 2021, and that will be the 10-week period would be May 30th, 2021 to August 7th. And this is, I think, a wonderful opportunity to apply. So it really would be wonderful if people apply for this program. If you want more information, please contact Solange Brown. Here's her email. You can also find it on our website. She's the co-chair for the Committee on Diversity and Inclusion. And she knows that I'm mentioning this today. So I think she'll be very upset with me if nobody emails her after today in the next week or 2. So please, send her email, even if it's just to find out more information about this program. She is a wonderful scientist, and she's also super nice, and she'll be more than happy to talk to you more about this summer internship program. That is the general overview. I wanted to leave time for the students to have some mention and also to answer questions. These are some of our students, and we're very excited, for students from UPR to join us. So we really would love you to apply for our program. And I will leave it there.
Thank you, Chris. So we still have a couple more minutes. I don't know if the students want to have questions for Giancarlo. There's another graduate student from.
Yeah, Lionel.
Lionel.
I think he's in the call.
So any questions for Chris or the students?
Yeah, so I had a question.
Uh-huh.
Dr. Porter, I heard that you mentioned summer internship opportunities, but I was just wondering if Hopkins also offered post-bac opportunities for us students who are seniors and graduating this year.
Yes, there are post-bac opportunities. There's one called PrEP. So there's lots of great opportunities. I would say go on the website. Our website has full details for all those. I think the one you're most likely interested in is one called PREP. So just check that one out.
Yeah, to give a little bit more information about that program, the deadline is going to be on February 15th. Normally, students spend 1 year doing research at Hopkins, but you have the opportunity to spend 2 years at Hopkins if you decide. So you're going to basically be paid a stipend. It covers also your benefits like medical and health insurance. And you will have an appointment like full-time doing research in a lab. And you will have also your own like mini thesis meetings. So a group of professors providing advice to you, professional training and professional development seminars. So definitely it's a great opportunity. Sorry for my neighbor's dog. There are a couple of students from the UPR that has gone through the PrEP program, so I'm more than happy to put anyone in contact with them for their experience.
Okay, thank you guys so much.
There's a question from Cielo. She says, how does the summer internship program requirements vary between students who are in different years?
Right, that's a great question. So what they're looking for when you're applying is that you've demonstrated some commitment or some desire to be a graduate student, that is something that's a driving need for you, and that you've shown that in some way. So if you're a freshman, it would be great to see that you've done some type of research experience or tried to get some kind of research experience in high school, for example, or even in your first year as a freshman. The reviews are done somewhat holistically, meaning that each application is evaluated based on its own merits. And they're looking for, if you say you really are interested in being a graduate student and want to get more research experience, how do you need to demonstrate that in some way? And which year you are at, you might be required, if you're more like a junior, you should have looked for some opportunities as a freshman and sophomore, for example.
Yeah, one of the things I wanted to raise this for you now, if you're a part of the admissions and things like that, with the pandemic, one of the major challenges have been that a lot of our undergraduate students have not been allowed in the labs, for example. Graduate students are, but undergraduates are not. In fact, in Puerto Rico, if you're a minor, they won't let you in at all. And so Kristina, you were saying?
What I was going to say is we're currently reviewing the applications for this year. And basically, that's something that holds for everyone. And it's just something that's spelled out in the reference letters. And we're reviewing graduate student admissions the same way, applications the same way that Chris just said, we're reviewing them holistically. So that's not going to be held against anybody. It's essentially clear that all of us have been working from home, that people have not been able to go into the lab, and that the research experience will look completely different. I think what I would encourage the students to do is to ask their main reference writer to describe that in their letter, and to maybe put it into their personal statement or into their CV, just to say, this is what I had planned, which is what I've seen in the application, saying this is what I have planned. I wasn't able to do that. So instead now I've done this other thing. And we all understand. We're in the same boat. Like, we haven't been running either, so nobody should feel bad about that. It's, if explain it, because then we understand even better, but we're not going to hold it against people.
Thank you, Kristina. That's very reassuring, because I just have these lots of my students, right, that they feel that they want to be in the lab, but there's so much you can do online, right? I mean, I have a rat lab, right? So I mean, they have to come and run the animals. They have to come and take, they have to understand how to inject a rat, how to do histology. I mean, so much for virtual reality there, no? So I appreciate what you just said, because that tells me that as expected, Hopkins is on top of things, and I appreciate that a lot for our students, because they are the ones that are all anxious, for their application, and they want to do a good job, and most certainly this is something that is very stressful. So I don't know. I wanted, before we go, both Lionel and Giancarlo, could you tell how much you like Hopkins? Do you love it there or not? No, don't mind Chris and Kristina.
Here, yeah.
So
we'll close our ears.
Yeah. I can talk first about it.
Yeah.
So I came into the conversation like towards the very end of Kristina's talk, so I don't know if anyone introduced themselves, but I'll quickly introduce myself. My name's Lionel. I'm a fourth-year PhD student in our department, and I'm in Dr. Carrie Martinowicz's lab at the Lieber Institute for Brain Development. I study mouse social behaviors, so if anyone's interested in that, you could feel free to reach out to me online or offline. But I love a lot of things about Hopkins. The things that drew me to Hopkins at first were very much both the research community and the social community that the students have. I think the students have a very, very strong support network that I was looking for as a graduate student when I was interviewing for programs. I really wanted both a community at which I would learn a lot from academically, but also that would grow with me during these very pivotal times in my academic career. And the students in the program are some of my best friends that I have grown to really love over my 4 years in the program. I would say it's still one of the strengths of the program. And the students have even done a pretty decent job of staying together as a community even during the pandemic. Of course, it's not perfect, but I look forward to being able to reconnect with everyone when hopefully this is all over. Another thing that I've really grown to love about the community is how approachable all the faculty are. The faculty genuinely do take an interest in becoming a part of students' lives if the students really want that. And I've become very close to several faculty in the department, have fostered really close relationships with them. They are both mentors and friends to me. And I know for a fact that that's not something that happens in every department. And I really value that about our department. Yeah, so if anyone has any questions about anything I've said, feel free to ask. But
can you share your email address?
Yeah, I'll put it in the chat.
Thank you. And what about Giancarlo?
Hello, everyone. Yeah, my name is Giancarlo Molina. I graduated from the UPR Rio Piedras in 2018. I actually went to Hopkins first as a summer intern in 2017, and I then stayed for PhD program. And actually, I'm conducting my thesis work in the same lab I interned during that summer. I'm currently working with Dr. Dwight Burgos in neuroglial interactions, more specifically looking at remyelination dynamics in the cortex. And at least for me, definitely, as you know, Puerto Ricans always looking for community and people to love. That sense of family when we are moving to the United States alone, it was definitely the major factor that I was looking for. And I found that on not only the neuroscience community, but also Hopkins at large, you get exposure and the possibility to make connections to other people in other programs, in other schools at Hopkins. So definitely that's something that has been very valuable to my experience. And Lionel's experience of how PIs are approachable and what Chris was mentioning about how collaborative the environment is has definitely been a major part of my progress on the research side at Hopkins. Just talking to my PI about what I want to do or what we want to do as the lab to move the field forward. It was very easy to contact people, not only in the neuroscience department, but also on the biomedical engineering department and other schools, even at other universities in Baltimore, that we can actually establish collaborations very easily and join forces together and basically take our expertises to make a collaborative effort. And it has been as easy as talking with the PI or some other postdoc on the hallway, on the neuroscience happy hours. And actually, one of my main collaborations for one of my aims on my thesis started in a conversation in a happy hour. So definitely has been a really great experience, very rewarding, those connections, long-standing. I'm currently on my third year in the program. I can definitely say that it has been very vital in my development as a researcher and as a person. So I'm currently living in one of the neighborhoods that Chris said, right next to the shuttle, and very easily to navigate around through the shuttle and just walking. I'm north downtown, so I can actually walk, have a 25-minute walk to the inner harbor, see water, that it's very important to us. So I have definitely loved Hopkins and all the experiences that I have got, not only at the neuroscience department level, but also at the university level.
Great. Happy students. That's all you need. Happy students and productive, of course, as well.
So I will send also my email. So if anyone wants more information about the graduate school or summer internship, or if you want to know a little bit more of the experience from other UPR students that went through the prep program, more than happy to connect you to. And if you want another pair of eyes through your applications for summer internships or the prep programs, more than happy to do as well.
Thank you. Thank you very much. Well, I think we are reach our time, a little bit over, but I just wanted to hear the students, of course, which are the best part. I would like to thank both Chris and Kristina really for your gracious efforts to be here, and for us, and for Giancarlo and Lionel for being as well. And for all of us here at the University of Puerto Rico, we thank you for being here. And this is our last BPN Doer seminar. And I don't know, maybe we'll do something next semester. I'm not so sure. Let's see if I still have a couple more brain cells to organize such a thing for another semester. But I think this has been a wonderful, wonderful experience for me. I mean, especially to be able to connect with students and faculty that are so committed for enhancing diversity and inclusion in the neurosciences. So I really appreciate your support on this. And so Merry Christmas, Happy New Year, Happy Holidays, and I hope you enjoy, have a safe one, and thank you. Gracias.
Thank you.
Bye.
Happy holidays.